The first one. When they established a pH gradient in half of the mitochondria and the other half of the mitochondria did not get a pH gradient. They were at pH 9 on both sides of the membrane. So what's the question they're trying to answer? Yes, sir. So you have to remember that in one of these treatments, you have the same pH across the membrane. So I'm going to say the same thing, that you have the same pH across the membrane. Here you have different pHs across the membrane. So you can't just tell me if they change the pH, because that alone does not explain what is happening in this experiment. Yes, sir, and then sir.
Does the pH in the outer membrane affect the ATP?
It's slightly closer to what you need. Yes, sir. Aha! So you have to remember, it's what you have across the membrane, not just the pH on one side of the membrane or the pH on the other side of the membrane. What they're doing is, do you have no pH gradient or do you have a pH gradient? So do you need the pH gradient to make ATP, or without the pH gradient, can you get ATP? And you have to remember, there is no electron transport here. Why not? They're isolated mitochondria, so if you don't give them pyruvate, there is no electron transport. Yes, sir.
So is it essentially also asking an artificial proton gradient outside the mitochondria is just going to force
the gradient is not outside the mitochondria. The gradient is across the membrane.
But will this proton motive force, like a higher concentration of protons outside, will essentially force the ATP production in the mitochondria?
Well, the whole point is you are providing a proton motive force, a proton gradient in half, and you're not providing it in the other half. So that's your independent variable. So that tells you what they're trying to figure out. So in the absence of electron transport, which is what you have in this case, because you don't have electron transport because you're not giving them pyruvate. In the absence of electron transport, is the proton gradient all you need? As chemiosmosis as proposed by Mitchell was that the proton motive force, the proton gradient, will provide the force to carry out work, in this case the work being synthesis of ATP. And so that was what Mitchell proposed in either late 50s or early 60s, I can't remember exactly when. And so this was an experiment. Actually, the first set of experiments were done in chloroplast, and you'll see those in a couple of weeks. And then later, the experiments like this were done in mitochondria to support Mitchell's hypothesis that the proton gradient can provide the force you need to do work, such as ATP synthesis. And so the question here they're asking is, well, no proton gradient, we have a proton gradient. Is that all you need to make ATP in the absence of electron transport? And what did their data show? Well, first of all, what would be the hypothesis for that question? At the question, it's easy to say what the hypothesis is. Sir? Stop saying pH outside. We're not interested in the pH outside. What are we interested in? The gradient across the membrane. You don't want to talk about the pH outside or the pH inside. All that matters is the difference. So there is no difference here. There's no gradient. Here there is a difference. There is a gradient. So now go ahead. So if the question is the pH gradient enough to make ATP, what would be a potential hypothesis? Yes, it is. And your null would be, no, it isn't. So if you have a question, it's kind of easy to answer to figure out what the hypothesis would be. Usually the hypothesis is yes it is or no it isn't. It's not always that simple, but for this experiment, it is that simple. Yes, sir?
What if the concentration of protons in the different pH 7 or 9 isn't the actual reason why there's what if the concentration of the protons in pH 7 and 9 doesn't really have to do with the gradient, instead, but it's that proteins don't function normally in pH of 7 or 9. What if it's the pH 7?
But the inside is pH 9 regardless, and that's where the protein is.
What if the shape of the protein changes in the pH of 7 or pH of 9?
Very good question, whether or not putting it in pH 7 on the outside affects the shape of the protein, and we don't know that at this particular point. That is true. But your independent variable is absence of a pH gradient, presence of a pH gradient, and all you can say from this experiment is in the presence of the pH gradient, we got ATP synthesis. In the absence of the pH gradient, we didn't. So this hypothesis supports chemiosmosis. You never say it proves. Those of you who kept saying prove, prove, prove when during the last exam, I crossed out prove every time you wrote it, because we never say Pasteur proved this, or Miller-Urey proved that. Stop saying that. They supported the hypothesis or they disproved the hypothesis. You do not prove a hypothesis. It's not a math class. In math, you prove hypotheses. In biology, you do not prove hypotheses. You support them. So here, you support the hypothesis that the pH gradient provides the force to make ATP. Lack of pH gradient, no ATP. And it might have to do with more than just the pH gradient, but you don't know that because the only independent variable you have here is the pH gradient. In the second experiment, what's your independent variable? What's different? Yes, sir? Presence of the ATP synthase. Half of the vesicles got the ATP synthase, the other half didn't. So that tells you which question they're trying to answer. Yes, do you need ATP synthase, this particular protein, to make ATP, or is the pH gradient by itself enough? Because the previous experiment showed pH gradient, you got ATP, but you had a whole mitochondria, so there are a whole bunch of complexes in the mitochondria that could have made the ATP, and so now they're isolating that particular protein. Do we need this protein to make ATP? Or is the pH gradient by itself enough? And it turns out, their data shows you need the protein. Yes, sir? It had been proposed because it was actually studied in chloroplasts before mitochondria. So from the chloroplast studies, they had suggested the term ATP synthase. Very good question. So if your question is, do you need ATP synthase to make ATP, what would be your alternative hypothesis? Yes, you do, and your null would be, no, you don't. And the data supports which one? Your alternative hypothesis, that yes, you do need that protein, because without it, you don't get any ATP. So far, so good? Yes, ma'am. So the first experiment showed us that in the mitochondria, proton gradient required for ATP synthesis. And the second experiment showed us proton gradient by itself, not enough. You need that particular protein, ATP synthase. All right, so we are going to spend a couple of minutes on fermentation. So what happens when there is no oxygen and the pyruvate cannot go into the mitochondria? Which means you're stuck with glycolysis. But there's a problem, because yes, glucose can be broken to pyruvate and you get 2 ATP out of that, and if there's oxygen, pyruvate can go into aerobic respiration, you get 30 something more ATP out of that. But if there is no oxygen, you're stuck with pyruvate, and then you run out of the other substrates that glycolysis needs. So you have to do something with the pyruvate. And that's what fermentation allows you to do. The only thing fermentation does is recycle the other substrates that glycolysis requires. So let's take a look at what they are. By the way, this is a figure from your textbook, and there's a little mistake in it. So, mistakes. Here's your glucose. Here's pyruvate. You should immediately see the mistake. Two ATPs. It should be making 2 ATPs, not 1 ATP. But that's beside the point. The other thing that you need in glycolysis is you need this molecule, NAD+, to make NADH. That's part of the reactions in glycolysis. If you don't send pyruvate into the mitochondria, you don't end up using NADH and you will not make NAD+. Well, you run out of NAD+ eventually. So somehow NADH has to be used, NAD+ has to be recycled. In aerobic respiration, mitochondria will do that for you. Without oxygen, no aerobic respiration, fermentation will do that for you. And fermentation can do it in different ways depending on which organism you're looking at. So you could have what we call lactic acid fermentation, where the final product is lactic acid, or ethanol fermentation, where the final product is ethanol. Both of them do the same basic thing, is use the NADH and recycle the NAD+. That's their whole point. They do not make any additional ATP, they do not make anything else useful. And lactic acid and ethanol are actually toxic byproducts. So you have to get rid of them. So when yeast make ethanol, they secrete the ethanol to the outside environment because that's a toxin. So if you enclose the yeast in a bottle, the toxin accumulates and you get your alcoholic beverage, which then eventually kills the yeast because they can't survive in their own waste. So after a while, you end up killing the yeast. So the alcohol concentration in your alcoholic beverage that you're brewing in whatever glass container or barrel you're doing cannot ever go above a certain level. So all of those cocktails that some of you might drink, those of you are old enough, that might have higher concentrations of alcohol, how do you get that? Distillation. So you might distill the alcohol to concentrate it. So if your vodka, which is, I don't know how many proofs these days people drink, that's distilled. That's not coming directly from the yeast. So here's the waste product. But the whole point is recycle the NAD+, otherwise glycolysis stops. And if glycolysis stops, what happens? You die, because not only do you not have aerobic respiration, you don't even have fermentation anymore. The only difference here is that pyruvate and lactic acid are both 3-carbon compounds, whereas ethanol is a 2-carbon compound. So as you make ethanol, you do release 1 molecule of carbon dioxide for each pyruvate. So here's another mistake. This CO2 should have a 2 in front of it, and that ATP should have a 2 in front of it. Which is the reason why when you use yeast to make dough for your bread, what happens to the dough? It rises because it accumulates CO2 inside the dough, and that helps the bread become fluffy. And when Passover comes around and some of you eat matzah, matzah is not fluffy because matzah does not use yeast to leaven the dough. Somebody's hand went up in the back. Yes, ma'am. So in the absence of oxygen, when yeast is working in your dough, what it will do is it will take the pyruvate from glycolysis and turn it into ethanol. So your dough actually will include some ethanol in it. And when you bake your dough, you evaporate the ethanol out of your bread. And the CO2 accumulates in the dough and makes the dough rise, and the bread is fluffy. So all those gas bubbles in the dough that show up as little holes in your bread. When you slice the bread, you see the little holes. That's where the CO2 was trapped.